We urgently need new medicines to treat respiratory tract infections. Viruses and bacteria constantly evolve, and that very fast, which makes treating them so hard. We have only a few, if any, drugs to treat viruses. And for bacteria, antibiotic resistance is an emerging huge problem. Researchers worldwide are looking into promising new approaches to treat respiratory tract infections. The problem is, however, we see a really wide gap between this research and the actual development of drugs towards the clinics. We at Fraunhofer ITEM aim to contribute to fight life-threatening respiratory infections with our expertise in translational research. With new disease models that mimic more closely what happens in the patient's lung, we aim to bridge the gap in anti-infective drug development.
We have the unique opportunity to work directly with human lung material obtained from surgeries that we cut into living lung slices. These are miniature lung pieces, which we can cultivate in the lab and use to mimic infection, investigate how the pathogen interacts with the host environment, and to test the efficacy of new drugs. For example, we use these slices in our new big international consortium ICARE, where we team up with experts from Medical School Hanover and Institute for Glycomics in Australia. We combine our strong expertise in target identification, inhibitor design, and pharmacology to explore novel anti-infective therapies against respiratory pathogens such as influenza. To do so, we infect the slices with influenza virus, treat them with different doses of new viral inhibitors, and evaluate how the new compounds are able to block the infection. We determine how the virus replicates in the tissue and how this affects tissue viability and the host immune response. In this way, we can profile new compounds and select the best candidates before going into proof of concept in vivo studies.
We are especially interested in bacteria that are able to adapt to the host and cause chronic infections, such as Pseudomonas aeruginosa. This bacterium is relatively harmless to healthy people, but poses a huge problem for patients with certain lung diseases. Often these bacteria also grow in biofilm, which is like a slimy layer that acts as a protective shield. Inside this biofilm, the bacteria can literally hide from the immune system and become unresponsive to most antibiotics. Using our lung slices, we investigate how the bacteria adapt to the host lung. Understanding these mechanisms is key to develop new drugs against chronic infections.
For efficient treatment of lung infections, local administration of the drug by inhalation is preferable, as you apply the drug directly to the place where the pathogens are located, so into the lung.
If we want to test the effect of inhalable substances on tissue, things become a bit more challenging.
So we invented a device specifically for this purpose. Using the Fraunhofer patented PRIT Expo Cube, cells or tissues can be exposed to aerosols or inhalable atmospheres in a controlled and efficient way. We currently establish this state-of-the-art device as a novel tool to test inhalable antibiotics. In this way, we want to reduce animal experiments in infection research.
A therapeutic concept that currently sees a strong revival is phage therapy. Bacteriophages are small viruses that infect bacteria, and only bacteria, not the host. That's why it's a promising natural weapon to fight multi-drug resistant bacteria. We are part of the Phage for Cure project, which aims to develop inhalable phage therapy versus the bacterium Pseudomonas aeruginosa. Our task in this project is to check if the phages after the nebulization process are really still active and kill the bacterium.